Joining us today on Superheroes of Science, we are pleased to welcome Sebastian Kenny. Kenny is a PhD candidate in the DOS lab at the Department of Chemistry in Purdue University. And he does research working with the ubiquitin system, or as he just told us, the kiss of death. So we are excited. Thank you for joining us.
Thanks for having me.
Yeah, it's I don't know. I'm a little nervous about this. I'm not sure what this system is, but to find out somebody's that we're interviewing researchers the kiss of death must be intriguing.
Yes.
So I'll let you explain so that we know. Right now, I'm just happy we're not in the same room because that makes me.
Well, okay, so the kiss of death, even though it's called the kiss of death, it's actually a good thing that we have that in our system, right? And so basically, just as a background, we have proteins in our cells, right? And proteins are important players in our system because they are pretty much the machines, the molecular machines, or the workers of our cells, if you may, right? And so you can think of the cell sort of like as Purdue University, for example, right? In Purdue University, there are people who works to manage all of the data. There are people that works as, the law enforcers, police officers and so forth, right? And there are also people that, mitigates risks and stuff like that, protect Purdue people and so forth. So same thing happens in our cells, right? We have proteins that does all of that work. So, you probably heard from other researchers that does work on other different proteins. They study specific things. Now, what happens is our proteins doesn't always stay good, right? And why do I say that? Sometimes they grow old, and when they grow old, or it doesn't have to be growing old. Let's say the protein gets a mutation from radiation damage, sunbathing for too long, something like that, right? What happens is these proteins get bad, right? And so then if they are bad and they still exist in our cells, right? The ubiquitin, or it won't be good for our cells, right? You could probably see why that is. What happens is our cells know how to do that, how to mitigate this. And so what they do is they tag this, rogue protein or protein gone bad, right, with ubiquitin. And what happens is this attachment of ubiquitin leads to so many different signaling pathways. And so when I say signaling pathways, one of them is the protein degradation pathway. And so it pretty much just, when you attach this ubiquitin or supply the kiss of death, hence where it gets its name from, right? The protein gets pulled to this protein in our cell, other protein, okay, called the proteasome. And the proteasome is pretty much the protein shredder. And so once it is brought over to the proteasome, right, that protein is shredded and it's non-existent in our system anymore. If, I give it in terms of analogy, let's say someone like a worker at Purdue University has gone rogue, right? What ubiquitin does is ubiquitin is, again, like the security guards or the police officers. They basically pull this rogue worker at Purdue University to jail. Is just keeping them there, but instead of just keeping it to jail, it's sort of like punishing them by the death sentence in a sense, so that they're not present anymore. But of course, you can see how that's very important in our cells, right? Because we need to regulate this good balance. So hence the name, the kiss of death.
So did you say that our cells will just on their own, they will tag these? I mean, that's like a normal part of our process, like of cells process?
Yeah. So great question. What our cells do is the attachment of ubiquitin, yes. The short answer to that is yes.
Okay.
Our cells have 3 proteins that sort of works. They sort of like relay ubiquitin. It's like a relay race, right? They pass it on from E1 to E2 to E3. The specifics of it, I can get into details with, but for the purpose of this, basically it's a relay between ubiquitin to ubiquitin gets activated by E1. And then it E1 transfers it over to E2, or E2 takes it from the E1. and then E3 takes it from the E2, or the E2 usually actually binds to the E3, and the E3 is what determines what protein in our cells needs to be ubiquitinated. And so there's so many different proteins in our cells, right? So the E3 actually acts as a specificity factor. It chooses specifically what do I want to ubiquitinate, right? And there's actually 600, and there's more discovered, E3 ligases or E3 enzymes discovered to date and ongoing. So yeah.
Okay, so if my body detects it's within each cell the things that aren't going right, and so protein's gone bad, I guess. But if it does it automatically, what is your goal? What are you trying to do with researching and studying this?
Right. Yeah. So in our lab, so in the DOS lab, we find this important, or like interesting rather, knowledge on ubiquitin. And recently, in the past 20 years or something like that, we've discovered more and more that pathogens, so when I say pathogens, I mean bacteria and viruses, right? They produce proteins that hijack the ubiquitin system. And so why is this important, right? The ubiquitin system is only present in eukaryotic organisms, meaning, us humans, plants, and so forth. These pathogens, bacteria and viruses, they don't have the ubiquitin system on their own, right? But it turns out they can still code and produce proteins that hijacks the ubiquitin system. Why is that important, right? And why is it even interesting? It's interesting because to think about it, right, you can think of these pathogens, bacteria and viruses, to be like thieves wanting to infiltrate. Again, I use the cell as like Purdue University, right? To infiltrate Purdue University, or the cell. Now, of course, we already have all of our systems in place, to make sure that nothing, ever harms us in a way, right? To keep our system going. And so pathogens know this, and pathogens need to look at, okay, what are the system in place that's in there that is present to kick me out? And if I know what that is, I will, do something so that I can work around it, right? And so what happens is with the ubiquitin system, when pathogens infiltrate our cells, right? Our cells know this, and our cells usually undergo this thing called xenophagy. And so xenophagy is when the cell kills itself, because if it doesn't kill itself, then this virus or bacteria can survive, and if they survive, then it can actually be more infectious to other cells and so forth, right? So what this and the ubiquitin system is in play with regards to this. What turns out to happen is, this pathogens, as I mentioned earlier, produces proteins that hijacks the system. So let me just give you some examples. Chlamydia, which is a common known STD, right? When chlamydia infects the cells, right, turns out it produces this protein called DUBs. DUBs are deubiquitinases, not this dub that people do these days. So deubiquitinases, what happens is, as the name suggests, it cleaves off the ubiquitin chain that is already built. So you can imagine that, for example, right, chlamydia attacks the cell, it invades it, right, and then the immune system needs to start kicking in, and so the ubiquitin, is being relayed and tagged onto the different proteins that it needs to be tagged on, right? Mm-hmm. What happens is chlamydia stops it by, okay, never mind, I'm just gonna cleave you off over there, and because I'm cleaving you off, then the signaling and the response doesn't happen, because it's like, oh wait, was that a false alarm? Are we not needed anymore? And so the cell decides to just stay in it's normal, as if there's nothing going on. Right. So it's sort of like a they are using the ubiquitin system, what's the idiom, like a wolf in a sheep's fur or something like that.
Yeah.
So yeah, that our research focuses on that. And so we just want to know how these proteins that they produce, right, to hijack the ubiquitin system, how do they work? And so some of the systems that I mentioned earlier is ones that I've worked with, Chlamydia being one of them. My thesis project itself is on human papillomavirus, because human papillomavirus also does also produces a protein that hijacks the ubiquitin system in one way or another, not exactly the same. And Legionella pneumophila is one that, a lot of people in our lab is doing. But it's, when we delve deeper into it, it's kind of interesting to see how these proteins actually work in so many different modes. I only described one, but there are hundreds, if not more, ways that they hijack the ubiquitin system. So yeah.
That's really cool.
That is.
It's I guess I never thought about the cells themselves having this being like a system of things working. They just seem too small and simple for that to me. And so it's like, I'm like, that's really cool. Then the fact that you're able to study one small part of that, I think is just absolutely amazing. What type of instrumentation and observation are you to be able to see the function characters within a cell? How can you go about actually studying that? Because to me, it kind of blows my mind a little bit. You'll have to help me here.
Right, of course. So one way that our lab studies proteins, right? So our lab is a structural enzymology lab. And so what that means is, whoops, my dog just unplugged my laptop charger. So our lab is a structural enzymology lab. And so being a structural enzymology lab, we are interested in knowing what the structure of these proteins are, okay? And so by knowing the structure, we can sort of know how the enzyme or the protein functions, right? It's sort of like, let's say, you try to visualize a protein and then it looks like a Pac-Man, for example, right? Then you can sort of probably deduce that, okay, that protein's probably gonna like fold into something or like gnaw on something, Of course, that's an oversimplification. It doesn't really work like that. But knowing the structure sort of gives you an idea on how the protein functions, especially because, there's been prior work and with proteins, when they fold, there's always different domains, right? And different domains just means different parts on the protein, okay? And if there's different parts on the protein, usually right now we can try to compare with known protein structures. If they look similar to it, or the sequence of the amino acid on the protein is pretty similar, then we can probably deduce that, oh, it does a pretty similar function. And if it does a pretty similar function, then at least we know how they inhibit. So with regards to structure, the way that we or we approach it is we use X-ray crystallography, which pretty much means us growing the crystals of the protein, and then we shoot it with X-ray lasers to get the electron density, all of that very heavy biophysics topic. And then we can deduce what, the protein actually looks like. Another way that, our lab is starting to get into, and I've been doing and trying to get my hands on more, is cryo-EM. And so cryo-EM is cryo-electron microscopy. And this is a technique that's, just been a rising star. It's simpler in a way than X-ray crystallography because you don't need as much sample. You don't need to purify as much protein. And all you have to do is you put them on a grid and then you visualize them. Instead of on a light microscope, it's an electron microscope. So you can actually see atomic details in a way. Yeah. So we use that, and then to study its activities, we use many different other techniques. So our lab is also a biophysics lab. I'm lucky to be part of the molecular biophysics training program, which is an NIH T32 fellowship. And we've really been able to study a lot of different things. So for example, like if we want to study how the effector proteins or the proteins that this pathogens produce interact with other proteins, we can use methods to measure their binding affinity, meaning how tight do they, bind to the other proteins in the ubiquitin system, for example, and so forth. Yeah, there's, a multitude of techniques that we can use, but I would say the main ones are really this structural methods, because we do want to see what the structures look like. I guess just to add to that, in a way, you can think of how beneficial this is in terms of drug development, right? If, let's say, for example, you want to target the specific proteins, so that you don't get as severe of an effect from the infection of like this pathogen, knowing the structure of this protein sort of gives you a hint on like where the weak spots are, right? It's sort of like if your brother or your sister is very ticklish on the sides, right? Then you can attack specifically over there, to weaken them or something like that, right? And so same thing with these proteins. If you know the structure, you know what it looks like, you can target them better.
Wow.
So
Now,
what is the scale? It sounds a lot smaller. Sarah, I'll let you jump in. I've already started. So it sounds a lot. I mean, I'm thinking in my head, oh, I remember seeing cells. I remember taking biology, and looking at plant cells through a microscope. But it sounds like you're looking at things obviously a lot smaller than that. What scale are you working at with those?
Wow. Very small scale. So it's interesting, right? The discussion of size is, proteins are so small. So yes, the cells are small when compared to us, right? And then the proteins in the cells are, of course, way smaller than that. It's like, I'm trying to think. It's in the nanometer scale.
Okay.
So like,
well, I don't expect you to know off the top of your head the exact.
Of course, all proteins are different. Ubiquitin being a very small protein, it's even in the sub-nanometer scale, I would say, probably. But the system that I work with is around 10 nanometers, which is a very small scale. But to put it in perspective, right? It seems very small to us. But then it's like if we zoom out from us, right? We really are also only small. Wow, this is very philosophical. We are really a small being in this, huge Earth. And, like in terms of the galaxy, they probably see the Earth and it's like, oh, what's that small thing, right? But really, we're in there. And then inside of us, there's the cells. And then the cells itself contains proteins and, all of this materials. It's a very interesting thing to ponder about.
That's a very, very insightful. When you're preparing your samples, now, would you, as a researcher, would you run the X-ray crystallography yourself, or actually like use the cryo-EM? I'm so fascinated by this cryogenic electron microscopy. And then do we have that here at Purdue, or are those samples shipped off and you receive those back?
Right.
That's a lot of questions. I wasn't sure which one it is.
So with the X-ray crystallography, right, we do everything. We grow our crystals and everything. But of course, to get, a very fine beam of X-ray, it's not something that's easy. We have an in-house source, right? But actually at Purdue, we're pretty lucky that we're only located 2 hours away from the Argonne National Lab, or the Advanced Photon Source. Mm-hmm. And so we usually, as a lab, of course, pre-COVID, would take trips to go over there and shoot our crystals over there. And so, the facility is free of charge for us to use, right? We just go there, we submit our samples, and then we shoot them by ourselves, with, of course, support from there, which is a great thing. With regards to the cryo-EM, we do have a facility over here. So we are actually one of the consortiums that's located in the Midwest. So, people from IU would also come to use our facility, and other universities too. We have 3 microscopes now, pretty, really, really good microscopes. And so I've just been going over there. After I prepare my samples, I freeze my grids over there. And so grids are pretty much, if you do light microscopy, we use cover slips and those, glass slips, right? With electron microscopy, because everything is in the small scale, you use these grids. Actually, I have a grid over here. So, oops, you see the circles over here? I hope they don't fall over. There, you see it?
Yeah.
Those are bitty things.
Yep, they're very tiny. And so those grids is what we apply our samples on. And then we put them on the electron microscope, and then the electron beam just goes, or like it goes through the sample over there so that we can visualize it. So, in a way, it is very similar to a microscope. It does the same thing, but of course, the system is pretty different. Instead of using lenses, you use electromagnets and so forth. But yes, we do have a facility for that, and we have really good support for that. So I've been lucky to just been practicing this skill over here.
Yeah.
Well, I paused because I didn't want to cut you off again, Sarah. I didn't want to go. Sorry.
Oh no. Yeah.
So what's next? What's next? Well, you said, I don't know how far you are. I know you're researching and trying to figure out the structures. And so what's the next step in this? Where are you going next?
That's a good question. So, okay, I do know what I want to do, I guess, in terms of career for myself. So I want to finish off with an MD PhD degree, meaning that I'll finish my PhD and then hopefully I'll get my MD somewhere after this. But I really do like research. I'm a talker, as you can probably see. And so the reason that I wanted an MD PhD is because I wanted that patient interaction, which I think is very important. I've been volunteering at the hospital, Franciscan Health over here. And just talking to the patients and other volunteers and the doctors over there, it sort of gives me life. Because work in the lab is fun, and you get to learn new stuff, but then it gets really repetitive because experiments don't always work their first time, right? Otherwise I'd be done and be finished with my PhD, but that's not the sweet case, right? It's very repetitive, and I need ways that I can also contribute and know that I am contributing in the real world, and that's why I'm volunteering. But anyways, so MD PhD is sort of the end goal, but I do want to work in a research institution or a teaching institution where I can practice medicine, teach classes if possible, and also do research. So that's sort of where the long-term goal is.
So in PhD, that's like, so you're wanting to be a specialist, a type of doctor then eventually?
Yeah, at some point, yes.
Okay, in specializing in?
That is something to discern about still, but I'm really looking at pediatric surgery. I love kids, and they also bring me joy. Their happiness all the time, even though they cry a lot too. That is something that is very infectious, just like this pathogens, right? And so that's I want to still have that joy. No matter what stage of life I'm in, I guess. So that's why pediatric surgery is like one of the things that I'm looking into.
So how often, I guess I know a couple of doctors, but I never asked them what they did before they actually had a practice. How often would a specialist start off like in a lab, like you're doing? You're in a chemistry lab, like I don't know how many, I want to say biochemistry, but I think it's a lot deeper than that. And so how often would a medical doctor start off in a lab like this doing research before they became?
I think the emphasis on research has been growing in medical schools everywhere, from at least how I've screen schools and everything, it seems that they always put an emphasis usually on their, third year where they do full research or something like that. Or during their stay, they have to, start working on a project. It's probably not as, intense as a PhD itself, because with a PhD, you really have only one thing to focus on, which is your research, right? With medical school, you have to do so many other stuff. So of course, I think there's always exposure, and people are aware of what they do. But then I'm actually not sure if I could answer, how many people actually does, a PhD just like me, or like how much does an MD PhD together or something like that. Yeah.
Okay.
I think that is such an awesome idea then, because it just seems like, with how personable you are, to be able to understand the research side and then put it in a way to explain it to your patients someday, that to help them understand what's going on. I think that could be very empowering for the patient.
Right. And so that's also one of the motivations on like why I decided to do a PhD first. I feel like if I understand the system, in its molecular basis and like, the whole details of it, it would make it easier for me to understand, like me personally, right? To understand what's going on, but also to be a better communicator to, the patients, or, whoever I have to explain the science to. Because as you know, explaining science isn't always the easiest thing, especially with the many jargons that are always thrown out there. So yeah.
Oh, that's cool.
Well, this has just been so exciting to learn about this, about the ubiquitin proteins and what they do as part of the cell, and then what you're looking at. We really appreciate.
Yeah.
We appreciate your time. This is really cool.
Yeah, thank you.